which is the capacity of the single cell to express the whole genome during the cell division. So, totipotency is the special type of character. Totipotency is the special type of character which is exhibits only in the plant cells only. These the capacity of totipotency, the cells are exhibits the new plant cells. The ability of cells change their metabolism, growth, and development is just as significant and essential for the regeneration of the entire plant. So this one is only to involve the total plant growth. The plant tissue culture technology is being widely used for large scale plant multiplication. So, these plant tissue culture is a technique mainly involved in the use of large scale plant production. In addition to being used to research, they know the essential for the plant propagation, disease eradication, and the generation of secondary metabolites. So, the plant tissue culture. The plant tissue culture is first of all introduced by the in 1902 is tried to cultivate the individual palisade cells from the leaves. So, in the salt solution, uh, sucrose for the month stored starch, but ultimately did not divide. He is the considered the father of plant tissue culture since his experiment set the stage of development tissue culture technology. So first of all, the Haberland. The Haberland is a scientist, plant physiologist. He is introduced the plant tissue culture. So, what are the conditions are involved in the plant tissue culture? So, the choice of medium is based on the types of plant species, mainly, which is the what type of medium is used for the growth of the plant tissue culture. So that only they are the depending on the medium uh, medium of tissue culture. Explants are used for the culture of optimal response. All the nutrients are required for the plant to proper growth and development should be present in the plant tissue culture media. So all the nutrition medias means all nutritional values, the essential microorganisms, micronutrients, macronutrients, growth hormones, plant growth hormones, like how gibberellins. These are nutrition for the in the tissue culture. Macronutrients, micronutrients, vitamin, other organic ingredients, plant growth regulators, carbons, a few gelling agents make up of the majority. All the depending on the plant growth, depending on the medium, these micronutrients, macronutrients, and vitamins, ingredients, other organic matter, and plant growth regulators. These are essential for the in the media preparation of the plant tissue culture. So, similarly, the hormone level and culture variable like temperature, pH, light intensity, and humidity also play an important role in the success of tissue culture. Temperature is also very essential, and pH, light capacity, and humidity also. These all aspects are very essential the preparation of the tissue culture technique. The mineral consists of macronutrients such as nitrogen, potassium, phosphorus, calcium, magnesium, and sulfur, and other micronutrients such as iron, magnesium, zinc, boron, copper, molybdenum, and cobalt. These micronutrients and macronutrients all are the very essential for the preparation of media which are involved in the plant tissue culture, and some vitamins also necessary for the health plant growth in the tissue culture. The vitamins like thiamine means this one we call as a B1, and uh, peroxidase means B6, and nicotine acid, other vitamins such as biotin, folic acid, ascorbic acid, and vitamin E, that one we call as the tocopherol. These are some added in the media formulation. Vitamins also B1, B6, and vitamin C. These the vitamin E is also. This one is a very essential in the plant growth. Okay, so all the essential for the plant growth. Okay. So now we are discussed the pH. The plants are require an external carbon source, sugar, the most commonly used carbon source, sucrose. Other sources used as glucose, maltose, and sorbitol. So carbon is obtained from the glucose only. The pH of culture medium remains vital as it influences the uptake of various components of medium and regulates a wide range of biochemical reactions. So most media are adjusted to the pH around 5.2 to 5.3. A higher pH may be required for certain cultures. So most of the tissue culture media, the pH range between 5.2 to 5.8. This is the most suitable for the growth. So plant tissue culture, the plant tissue culture media, the most popular media for in vitro vegetative propagation of the various plants in Murashagi and Scoop medium. That one we call as MS medium, Murashagi and Scoop medium. So these is the name is given by the Murashagi and Scoop. That one we consider as MS medium. For the culture is either solid or liquid medium can be we can use. And macrons and woody plants media. This is also there. And plant tissue culture growth regulators. These are the PGPRs, means plant growth regulators are the crucial for determining the development of plant cells and tissues in a culture medium. plant growth hormones, either regulators. These are the crucial means very essential in the plant tissue culture. The most commonly used plant growth regulators as which are the auxins, cytokinins, and gibberellins. Mainly auxins and cytokinins. Sometimes gibberellins also very little amount of gibberellins also we are using. And high auxins concentration often favor the development of root. The most commonly used auxins are IAA, indole acetic acid, indole butyric acid, naphthalene acetic acid. So sometimes 2,4-D, 2,4-dichlorophenic acid, 2,4-dichlorophenic acetic acid. That's the short name 2,4-D. These auxins are used in the tissue culture. And cytokinins. So cytokinins, which is the promote the cell division and shoot growth. The most commonly used cytokinins, that is the BAP, means benzylaminopurin and zeatin, isopentenyl adenine and kinetin. These are the used cytokinins in the media preparation. So cytokinins are generally dissolved in dilute HCl or sodium hydroxide. A clump of undifferentiated cells called as the callus, which is developed when the auxins and cytokinin levels are balanced. So undifferentiated cells means mass of the cells are developed during the tissue culture where the plant growth hormones means auxins and cytokines are in the balanced stage only. That one we call as a callus. There is no differentiation of the plants means that one is the mass of cells. That one we call as a callus. So another one is the plant tissue culture vessels. So another, the critical aspect in the plant tissue culture is the management of the gaseous hormone ethylene. This ethylene is very essential and very dangerous gaseous. In closed culture vessels are used in vitro plant growth to ethylene build upon often determine the culture. The addition of ethylene biosynthetic inhibitors such as the silver nitrate. Okay, sorry, some internet issues are there in the So the cultures are grown involved in growth rooms and growth chambers. Humidity, light, and temperature must be controlled for the proper growth of culture. So there is a 16 hours light photo period is optimal for tissue culture, and the temperature of 22 to 25 degrees is used for the most laboratories. That is the room temperature. And the cool white fluorescent lamp also supply in the light intensity, and relative humidity around 50 to 60 is the maintain the growth chamber. Some cultures also incubate in the dark condition. So cultures can be cultivated in various containers, including test tubes, glass, petri dish, and some bottles also, depending on their feasibility. Different types they are to use. So next aspect is the sterility. How we are to sterilize or sterile the remaining all the environment autoclave. The preservation of the sterile environment is necessary for effective tissue culture. The laminar flow hood is used for the all the tissue culture work. A dust filter and high efficiency particular air filter are used in the laminar airflow. The surface of the plant tissues naturally contain a variety of bacteria and fungi. Before the tissue culture, it is the crucial to thoroughly clean the explant since the contamination contaminant can proliferate in the culture media. They also compete with the plant tissues for the nutrients retrieving in those nutrients. The bacteria and the fungi can rapidly surpass the plant tissues and destroy them. The explants are commonly surface sterilized using sodium hypochlorite and ethanol or some fungicides when using the field growth tissues. So the type of tissues usually decided the time of sterilization. Leaf tissues is required to shorter sterilization than the seeds. It is the hard seed coat is there, so that's why they have the more sterilization time. They require the more sterilization time. Okay. So this one is the sterilization process means all the environmental should be sterilized and aseptic condition only. So coming to the tissue culture types, so types of the tissue culture here we are the different tissue cultures are there. That is the callus culture. First one is the callus culture. What is the callus? Already we are to discuss that one. Means undifferentiated cells. When we are to use it in the tissue culture, the explant after transplant the media and we have to maintain the auxins and cytokinins in balanced stage. Here, the undifferentiated markers appear that one we call a. So now we have discussed about the how callus will culture callus. Recognize levels are wounded. When the explant is cultivated on media that promote the development of undifferentiated cells that we call as a callus is formed. So the maturity cells are formed with the auxins and cytokinins. Using the plant growth hormones, callus can multiply continuously or be directed, develop organ or somatic embryos. This is the callus culture. Uh, Only develop uh, uh, from that undifferentiated mass of cells when we are to add the auxins and cytokinins. It can be multiplied continuously and to develop as organ of somatic culture. Cell suspension culture. So the small fragments of loose gabriel callus can be cultured suspension culture in the medium. Cell suspension uh, bath culture grown in the flask for long periods. A portion of can be transformed in liquid medium to in cell callus and suspension. Culture can be cultivated from culture of suspension. Cell is generally higher than the type is the okay anther or microspore culture. So microspore, you know that one. So microspore is nothing but the pollen grain. The culture of anther are isolated microspore to produce the haploid plant that is known as anther culture or microspore culture. The anther is directly to produce the haploid plants. You know that one. The microspore is the male gamete, nothing but this one is the male gamete to produce the haploid plants only. The embryos can be produced via a callus phase or to be directed encapsulation of the development stage is characteristic of zygotic embryos. So compared to traditional breeding method, microspore culture is enables the creation of homozygous plants in a very short time. These homozygous plants are useful to tools in the plant breeding and further genetical studies. This one is the culture, microspore culture or antheric culture. Microspore culture, anther culture is mainly involved in the applied plants production only. When in the genetic engineering these required character or desirable characters they are want to they are choose these anther culture or microspore culture. So next type of the culture is the protoplast culture. Protoplast culture means the protoplast contain all the components that all the components So, protoplast culture here. The protoplast contain the all the components of plant cell except the cell wall. So each cell they have the cell wall and which is covered the protoplast. That is the cell sap. We also call sometimes protoplast and cell saps. So these protoplasts to protected by the cell wall only. Here without cell wall they are called as a protoplast. So protoplast can be used to create a somatic hybrids and regenerate whole plants from the single cell. So this one is the somatic hybridization. Mainly without cell wall, the protoplast fuse to form the diploid cells. This one is the regenerate the whole plants also. The cell wall of the explant can be removed either mechanically or enzymatically. The cell wall is removed before the uh, culture. So, with the help of the mechanically or the enzymes also used for the digest these cell wall. The protoplast can be cultured either in the liquid or solid medium. The protoplast embedded in the protoplast embedded in alginate and matrix and then the culture on a solid medium have a better success rate of the regeneration. Although protoplasts appear to be a very appealing method for the regeneration plants and transferring the genes, they are extremely delicate. So protoplast culture is very essential in the all the aspects. That means in the methods are regeneration and transferring the genes. So next type of the culture is the embryo culture. The embryo culture is very essential in the plant tissue culture to produce the disease resistance plant and desirable characters and some exotic characters. It is the technique in which the isolated embryos for mature ovules or seeds which are cultured in vitro culture. The embryo is isolated from the ovules or either seed only. That one cultured in the aseptical condition to produce a, a new plant or sometimes organ also which is produced. For species, those seeds are dormant or resistant or prematurely sterile. Embryo culture has been used as a helpful tool for the direct regeneration. We know that one. Some seeds are they exhibit the seed dormancy and premature condition, some resistance. This time only means which are used to culture the embryo. So the embryo culture directly involve the production of new plants. The in vitro control the population and fertilization to ensure the hybrid production. Additionally, the direct somatic embryos and embryogenesis callus can be produced from the immature embryos. Indirect means direct somatic embryos are embryogenic callus. can be produced the immature embryos also. Another one is the meristem culture. In the meristem culture means you use of the shoot only the tip is the shoot tip is have the meristematic tissue. What is the meristematic tissue? Just we are to discuss a few words about the meristematic tissue. So the meristematic tissue only involve the continuation divisions during the mitotic division. The meristematic tissue only to involve the continuation cell divisions. So that's why this one is the very small and very active cells are present in the meristematic tissue. Here only. So using the apical meristem tip, it is possible to produce disease-free plants. When we choose the explant, that explant is always disease-resistant plant only we are to choose. Okay, disease-resistant plants only we are to choose. So this technique means meristematic or can be for the meristematic culture. So meristematic tip culture or shoot tip culture, depending on the actual explants used when we use the tip of the shoot apex. The plant apical meristem make a good explant for the cultivation of virus-free plants. Hence, this method is usually to use the eliminate the viruses in many species. Already we discussed that one. That is the to produce tissue culture only. So when we are to the ways only one is meristematic tissue. Meristematic to choose the tip of the fruits to apex very suitable for the growth of the remaining all organs. So next one is the also clear, right? Okay. So coming to the regeneration methods of plant tissue culture. What are the regeneration methods are there? Which is include two methods are there. That is the organogenesis. Another one is the somatic embryogenesis. What is organogenesis? Organogenesis means to produce the organs. See here we are to discuss in the detail about the organogenesis. In plant tissue culture, it is refers to the formation of either shoot or root, the equilibrium of auxins and cytokinins, and the tissue's capacity to react to the phytohormones during the culture are key factor in vitro organogenesis. Depending on the auxins and cytokinins. These are the main in the root formation as well as shoot formation. So auxins, this is the regenerate the roots only. Auxins are
Induced or promote the roots, cytokinins, which are induced the shoots in the tissue culture from that explant or either callus. So this is equilibriumly they have involved the formation of the organogenesis. So here in vitro organogenesis, which is the two types are there: direct organogenesis, indirect organogenesis. So indirect organogenesis involves the formation of organs indirectly via callus phase. So the involves the formation of organs. This is indirectly through the callus only. For the production of transgenic plants, the induction of plants through the callus phase has been used. Okay, so I shown to the some tissue culture steps are there. So here diagram is also there. Just you wait. I show to. I am shown to these steps are involved in the see here. So these steps which are used in the okay, clear. These steps all the steps are there here. So means when we are to choose the plant, that plant is always in the aseptically and diseased free plants we are to use. So that part we call as explant. So the explant can transfer into the medium means agarose medium. So agarose medium. So after we have to add the growth hormones or some regulates auxins and gibberellins, cytokinins like that hormones. We have to apply it. That is to develop as a callus. That callus to develop as a plantlets, tiny plantlets. So that plantlets to develop in the transfer into the acclimation, acclimatization. That one we call as. So this is the technique involved in the okay. So what is the here organogenesis, indirect organogenesis, direct organogenesis? We are detailed. We are to discuss that one. Indirect organogenesis means involve the formation of organs indirectly via callus phase for the production of transgenic plants. Induced plants through the callus phase has been used. Either the callus is transformed, plants are regenerated, or the primary explant is transformed and the callus is formed, and then the shoots are cultivated from the explant. It is the more important for the transgenic plants production. Indirect method. Okay. So next one is the direct organogenesis. The direct organogenesis, which is involves the direct bud or shoot formation from the tissues without the callus stage. The plants are usually propagated by direct organogenesis for the improved multiplication rates and production of transgenic plants, but mainly for clonal propagation. So these direct organogenesis, which is involve the shoots or buds, flower buds, we have to use in the tissue culture. The plants are propagated as a direct organogenesis. Next one is the next tissue culture technique is the somatic embryogenesis. Organogenesis, another one is somatic embryogenesis. Okay, so here two methods are there. One is the organogenesis and another one is the somatic embryogenesis. What is the somatic embryogenesis? Here, in the somatic embryogenesis means, is it clear? Is audible? Hello? Okay. Uh, so that is the somatic embryogenesis in the means here. The somatic embryogenesis is the non-sexual development process that produces a bipolar embryo with a closed vascular system from the somatic tissues of the plant. So, in these non-sexual development, so it has become one of the most powerful techniques in the plant tissue culture for the mass clonal propagation. The somatic embryogenesis may occur directly or via the callus phase. Maybe directly, we are to develop from that parts or organ or either callus phase only. For the clonal propagation, direct to somatic embryogenesis is preferred since there is the less chance for the somoclonal mutations. Sometimes they are to appear the somoclonal variations. The indirect somatic or embryogenesis is usually used in the select of the desired. Okay, desired it's one is used desired characters. What are the desired are to okay. Uh, Now, again, we are to discuss here uh, routing of shoots. Another is there. So, before that one, the encapsulated somatic embryos are known as synthetic seeds. When we are to develop the somatic embryos, the somatic embryos is the capsule is there by the alginic acid. Sometimes calcium or alginate or alginic acid capsules are there, which we call as the somatic embryos or synthetic seeds. The synthetic seeds have multiple advantages. They are easy to handle and they are potentially to be stored for a long time, and there is a potential for scale up and low cost production, which is prepared very low cost. Okay. And another one is the rooting of the shoots. From the shoots only, here roots are developed. How they are to develop the rootings, we are to discuss detail. The successful acclimation of a planted greatly depending on the root system production. Okay. The rooting of shoots can be achieved in vitro or ex vitro culture. So after rooting only, the shoots are successful development as a plant. If the roots are not available, they are not to fix on the soil and to observe the nutrition or either water, whatever it, mineral or micronutrients, macronutrients. They cannot observe without roots. So, ex vitro rooting involves the penetrating the shoots with the phenols or auxins and then planting them directly in soil under the high humidity, which is significantly lower cost of manufacturing. This technique also allow the simultaneous acclimation of the rooted shoots. Means here the auxins already we discussed that one. Already told the auxins when we are to auxins are used the callus, the callus to show the rooting. So in vitro rooting consists of rooting plants in exenic condition. Despite the caster factor, in vitro rooting is still a common practice in many plants. That is, the rooting is very essential. Several factors are known to affect the rooting. The most important factor is the action of endogenous and exogenous oxides. So phenolic compounds are also known to have the stimulatory effect on the rooting. Already we discussed that one. The some factors also affected for the rooting, endogenous and exogenous, and some phenol compounds also stimulate these rooting factor. So chloroglucinol is a root promoter, is reported effect to root development, and catechol is a strong reducing agent, has been reported in the regulate IAA oxidation. So next one is the acclimatization. Acclimatization. What is acclimatization? Acclimatization means they transform the plants into the field. So once plants are generated by tissue culture, they have been transferred to the greenhouse or field. The process we call as acclimatization. Acclimatization is the final stage this in the tissue culture technique. This is requires that the plants be handed up before the transfer to the field to reduce water loss during the acclimatization. The plants are initially transferred to greenhouse or growth chamber. The relative humidity outside the vessels is often significantly lower than the humidity inside the vessels. Once the plants are acclimatization under the greenhouse condition, they are ready to transfer to the field. So in that acclimatization, this is the final stage when the plantlets are growth in uh, the tissue culture. Will after some days or 1 or 2 days, then which is shifted to the in the field or either greenhouses. Okay. So next, coming to the what are the advantages? What are the applications of the tissue culture? Here mainly the totipotency, nutrients, metabolism, division, differentiation, preservation of the cells. This one is the totipotency is a special type of character, which is the shows in the plant cells only. So during these totipotency only, they are the produce uh, the different types of the tissues. The morphogenesis and plant regeneration from the individual cells or tissues through the organogenesis or somatic embryogenesis. The variations were generated through in vitro culture. So here already we discussed that one, some variations are occurred. Another one is the evaluation of applied plants through the anther or pollen culture, including sometimes ovule culture also they are undertaken. So applied plants is the very essential to producing the plant breeding techniques. Okay. So in the applied plants only to develop from the through the anther culture, either pollen culture, sometimes ovule culture also. So hybridization program through the ovule, ovary, and embryo culture to overcome some pre-zygotic and post-zygotic sterility mechanism. So here, zygotic and post-zygotic sterility is also overcome through the with the help of the ovary and another one is the micropropagation of plant material. And another application in vitro selection of the mutants tolerant to biotic and abiotic stresses. Another application that is the in vitro culture and secondary metabolites biosynthesis. This is a very special aspect to synthesis of the secondary metabolites. And another one, the plant genetic engineering using the DNA transfer and in vitro techniques. So disadvantages of the plant tissue culture. In the disadvantages, what are the disadvantages here? In the labor intensity and expensive process, and the another one is the environment factor also vulnerable to many environmental factors. Okay. So, this is the tissue culture, and what is the definition? And some steps of media preparation, some steps are involved, and types of tissue cultures, types of tissue cultures, and what are the applications and some disadvantages also. Okay. So here, what is first of all introduce? Just we have to briefly we have to discuss about these introduction and types of here. The tissue culture. What is the tissue culture means? The definition of tissue culture, which is the in vitro aseptic culture of the cells, tissues, are either whole plant under controlled nutritional and environmental conditions to produce the clones of the plants, are either offspring of the plants, and that is the technique we call as a tissue culture. So how they are during the totipotency. The totipotency character, which is the capability to produce the new plants, the plant cells, that only uh, the, the cells only, the means plant cells only. Okay, so the Haberland, the tissue culture word or tissue culture technique is first of all introduced by the Haberland only. The Haberland is a scientist. Okay, is a scientist. Is introduced the these tissue culture. The remaining some different medias also there, and conditions here aseptical conditions, pH and micronutrients, macronutrients, vitamins, mm, environmental condition, pH, humidity. This is all already discussed that one. So MS Media, first of all, introduced the media by the Murashik and Scoop. So that's why we call as MS Media. And culture means cell culture, cell suspension culture, anther culture, and embryo culture, ovule culture, meristematic culture. These are the some different types of the cultures. Okay, so this class will complete, and tomorrow onwards we have to discuss another class. So thank you everybody. Thank you for thanking to attend the classes. Thank you so much.